Hello, everyone. Good evening. I hope you can hear me. If you can hear me, please type yes so that I'm sure I'm audible. I'm sorry for the issues, but we just hope that as we get along, the network becomes steady. So if you can hear me at any time, you cannot hear me, please just type not audible so that we could see it from our comments session here and try to um, update as much as possible. Okay, so I specifically thought of having this revision time with you for for microbial genetics, so that you at least you have an edge and have a clue of how to tackle questions on this aspect of the course. All right, so without much delay, we just gonna. I just want to start right away. Now we started with amino acids, even though that's not where we're supposed to start. So because of that, I want to pretty much start from where I was supposed to begin. How do I get this out of here now? How do I get this out? Okay. Okay. On the OBS. That's fine. Are they giving me a yes there? Hello? Reduce the fan. I'm actually waiting to know that you are hearing me, please. Are they responding? Yeah, you normally see comments now. I need your response if you can hear me. It's not. It's not showing green anymore.
Let's continue
Okay. Hello. Let me ask them on WhatsApp. Okay. Central dogman of molecular. Okay. Central dogman of molecular biology. And when we looked at this, we looked pretty at the structure of what it's all about, like a summary, and we said it's all about DNA. In case you missed this part of the lectures, you are having it now. DNA can be transcribed to RNA. And RNA can be translated to what? Proteins. Alright, so DNA can be replicated in this format, and what you have right here is replication. Then you have right here your transcription and your translation. Alright, so in the central dogman, RNA can actually be what? Transcribed back to DNA using the reverse transcription method. And of course that reaction is catalyzed by the enzyme transcriptase. Okay, and we said around this place that the DNA is a hereditary material. Okay, that is, it is the blueprint of life, and it's what makes a person unique from another. Okay, understand that this is a revision, and we'll just pretty much run through the process because it has earlier been taught. Okay, so DNA is the hereditary material. It is the blueprint for life. It is what makes one person unique from another. As such, DNA have the ability to make copies of itself. So when DNA is making copies of itself, right here, we do that in red. So when DNA is making copies, of itself, we say that is what? Replication. Also, it has the ability to transcribe itself into RNA, which is this step right here. And we also saw that it can therefore also translate into what proteins, which is the ultimate goal. So the ultimate goal is to what translate the RNA into proteins. That is what the DNA does. So we touched a little bit on nucleic acid, even though you told me about that you were being taught, or you've been taught a bit of it. So we looked at nucleic acids, what they are made up of, and we said that nucleic acids we said that nucleic acid are made up of purines, pyrimidines, nucleosides, and nucleotides. So these are basically what nucleic acids are made up of. And we say that the purines are made up of the purines in itself here are made up of guanine, adenine, okay, and we have the pyrimidines, let me take this a little up. Good. So we have pyrimidines are made up of cytosine, uracil, and thymine. So you should pretty know that of nucleotide. Cytosine, uracil, and what? Thymine. Okay, so it's important that you know their structures like I gave to you. Alright, then you now this thymine, you could have it abbreviated as what? CUT, where you have C for cytosine, U for uracil, and T for what? Thymine. Alright, and in their structure that I gave, their group in the 12 o'clock position is what determines a purine molecule. Why pyrimidines are a single ring, like you can see right here. Okay, so I'm looking at I'm giving you right now the structure of the purine adenine. Alright, so you have here Okay, so what we have here is your adenine, and in this other one, this has been deaminated, and what you have is this. Are there people there?
Somebody is saying I got from intermediary.
You said what?
Somebody is asking I got from intermediary. Is this one still in biotechnology now? No, no.
Which class is here? Is this person? I hope I'm not in the wrong class. The person writing that is in a different class. Hope he has not joined. I did not send the wrong link. Let the person know that we are doing a revision for uh, microbial genetics, and if that is not discussed, it's not yet time for their class. Biochemistry students are to come up by 1 a.m. So this 2 year actually represents purines. Pyrimidines, like I told you, are what? A single ring, and you have them like this. So by now you should have familiarized yourself with this structure. So what you have here is cytosine, and sorry about this. I'm pretty in a rush now because I have another revision time for 1 p.m. So it's my hope that we are able to cover as much as we can with the revision here. So this is uracil, and we have a bond here, and we have thymine that is methylated, and that is what thymine. Alright, so that's basically the structures, especially for this this aspect is really on the surface for you. Okay, so we now look at the differences between nucleotides and nucleosides. Okay, I know I specifically gave you that difference between nucleotides and nucleosides. Alright, so we said that nucleotides oh dear, don't know what's happening this morning. Alright. Okay, so we said nucleotides is the combination. It is the combination of a base, a sugar, and a phosphate group. Alright, a phosphate group. Is that simple? Okay, so mathematically, nucleotides can be said to be equal to a base plus sugar, okay, plus your phosphate group. Then this is different. This difference, when asked the difference between nucleotides and nucleosides, they are always often mixed up. You see students giving it in the opposite direction. So nucleosides is I need to be balancing my hands. So nucleoside is the combination, it is the combination of the sugar, and a base, without the phosphate group. So mathematically you can write nucleoside as just the base plus the sugar. Alright, so that's basically what we were able to cover with nucleic acids. We did not go much into other aspects as expected. Alright, so we now began to look at amino acids. Which I'll just begin to look at from what we've done previously. So we looked at amino acids, and we did mention that amino acids are the building blocks of what? Proteins. That's what we established. That amino acids are biological macromolecules. They are abundant and they are the workforce of what? Physiological systems. And that they are common, there are 20 common amino acids. We established that makes up the majority of proteins. What a specific particular amino acid is. So we also have the hydrogen atom right here. As part of the general structure, you have your hydrogen and you have your amino group right here. You have your amino group right here. Then we also have right here is what? Your carboxyl group. Right here is your carboxyl group. Okay, so that's what the amino acid generally signifies. Okay. Okay, so that's what the amino acid general structure signifies. Okay, so until you are asked to give the specific structure, amino acid is generally made of this structure. And of course, any specific structure will follow this pattern. Alright, so we went into the classification and we said that they are made up of 5 classes, which you can see here: the non-polar arrow group, aromatic arrow group, polar arrow group, positively charged, and what? Negatively charged arrow group. Alright, so that's what the amino acids are made up of. Alright, so we'll just move on. Okay, so right here we began to look at each of the classes. So at the end of the day, in looking at each of these structures and classes of amino acids, you can easily tell that you will be able to cover the 20 common amino acids. So for the non-polar arrow group, we have glycine, and you want to look at glycine, at the arrow group in glycine is hydrogen. Hydrogen is the arrow group in glycine. Then alanine, you have alanine as CH3. So you see how the arrow group of glycine is different from alanine, and that is how they all differs among the 20 common amino acids. Alright, I just want to be sure, are you still with me? Can you hear me? You can type a yes so that I will be sure you are with me. So we'll move right along. So still on the non-polar arrow group amino acid, we begin to see things like your leucine, isoleucine, methionine. Alright, methionine. They are actually the non-polar arrow group. They are represented that way, starting from glycine. Okay, so worthy of note in this amino acid is ability to understand the chirality of amino acid, and it's their chirality that enables them to exist as an enantiomeric pair. Their chirality enables them to exist as an enantiomeric pair, and among the 20 common amino acids, they all exist as enantiomeric pair except glycine. So why is it that glycine do not exist as enantiomeric pair? Okay, so we'll, I'll show you that briefly. Why? Okay, so let me just look at that in the structure of glycine. So right here is glycine, this structure here. Okay, so if I bring out this structure, so if I decide to draw the glycine structure right here, you're going to have this. Just like your structure of amino acid, you have your carboxylic group attached to your alpha carbon, attached to your hydrogen atom, attached to your amino group, and glycine in itself is a H. The arrow group here is a H. Is this H here that makes it the only amino acid that does not come in an enantiomeric pair? Why? Because it does not have a chiral molecule. It does not exist as a chiral molecule. Remember that we said that a chiral molecule exists when they have different substituents bonded to the alpha carbon. But all other 19 of the 20 amino acids exhibits that chirality, and hence they are enantiomeric pair. They have an enantiomeric pair. That means they are mirror images of themselves. They are superimposable, and they have isomerism. So they have isomers. Now for glycine, you have the same H hydrogen bond that is bonded to the alpha carbon here, bonded here. As such, this is what makes it not to exhibit as what an enantiomeric pair. Okay, so that is very key to structurally know what the difference, okay, and why it does not exist as enantiomeric pair. Then we'll continue looking at other classes. We just saw the complete class of the non-polar arrow group. The next class that we want to look at is the polar arrow group. The non-polar arrow group. All of these are under the non-polar arrow group. Okay, let's see what the next class is right here. Okay, so what we have here is the aromatic arrow group. So by aromatic arrow group, you can see the arrow group right here. So all of them, of course, follow their general structure, and their arrow group is what determines their specific amino acid. So when you say an amino acid has an aromatic arrow group, it means that it has a benzene ring attached, okay, an aromatic ring attached to what the attached in the arrow group, as part of the arrow group. So this is phenylalanine. Now the difference between phenylalanine and your tyrosine is your OH, and of course you can see the whole of this as a clear difference in your what tryptophan. So that is for the um, aromatic arrow group. There are just three of them. So we move on to the next amino acid grouping, and that is what your positively charged arrow group. Okay, and there you have your lysine. Okay, so you can see the arrow group well circled for differentiation. So you have, for positively charged, you have your lysine, your arginine, and what your histidine, as very key and worthy to note. Okay, then you move on to what your negatively charged. So from knowing the general structure for each group is just to know what the arrow group looks like, and you're pretty much good to be able to draw out any of these classes of amino acid. Then we went forward to look at the negatively charged arrow group. Negatively charged arrow group at what? pH 7. So we have, for negatively charged, they are just two arrow groups, and they are what? Aspartate and what? Glutamate. Aspartate and glutamate. And this is actually directly related to some uncharged, the polar arrow group, where you have molecules like asparagine and glutamine. Alright, so I may just as well draw that asparagine and glutamine here, so that you could see the relationship in structure. Okay, so your negatively charged arrow group, it has another carboxylic group at the end and just a methyl group for aspartate, while your glutamate has two methyl groups attached with what? Another carboxylic group outside the general other structure of amino acid. That is basically how to differentiate them. Then, with relationship and similarity to uncharged but polar arrow group, specifically for asparagine and glutamine, you want to see the relationship. Let me just do that in red here. So you have this, it's pretty close. So let me move it out a little bit. So we have aspartate, so I'll start with asparagine here. So it's almost, the similarity is just too clear. Almost the same structure, but what you see next is what actually differentiates them. So you see that it has this double bond, and instead of this O minus right here, okay, for asparagine you have what? An amino group attached. Okay, and that amino group is what differentiates it. Now the all of this is the arrow group, but this structure right here, this structure, right here, okay, is normally, okay, referred to as what? The amide of aspartate. And that's because it has an imine group attached instead of what? The O minus. Okay, so this group is actually under the uncharged but non-polar group. I just brought it out now because of what? The similarity. There are two that are specifically similar in this regard. They are like this: instead of aspartate, the amino acid name is what? Asparagine. Okay, asparagine, the abbreviation is this, and the later form is this. Okay, so if I want to do the other one, let me do it right here, which is your glutamate. You have CH and H3. Like I said, everything we follow is pattern. So if you are able to draw the structure of the negatively charged amino acid, this, at least for these two non-polar but charged group that has close similarity, is just almost the same structure, except that you replace the O minus with what? An NH2. So that's what we're about to see right here. So here we have NH2. So again, this is the arrow group, all right, and okay, all right, so this is the arrow group, and right here is what? Your what? Amide. Amide of what now? Glutamate. So amide of glutamate, replacing amine group, replacing what? Your O minus. This, like this, instead of being called glutamate, is called what? Glutamine. Glutamine, amine, asparagine. Okay, so that is pretty much it. So here you have your structure here and your Q. Okay, so that's basically that I thought to bring at that difference. And of course, the next uh, structure you want to see is your uncharged, okay, which I said, okay, this is what we just saw now: amide of aspartate and glutamine. These are not the only two that are polar group but uncharged. We have things like serine, threonine, and what? Cysteine. Alright, so that is very clear going forward. Now we want to look at other aspects, some things to know about amino acid, like we saw, and we saw some characteristics that we saw in class about amino acids and how to identify them. Okay, so we'll just quickly look at that here. So we say some things we wanted to know about amino acids, and we looked at what? Cysteine, for example. Alright, we saw how cysteine can be converted to what? Cysteine. Those are special features to note about amino acid. So we just quickly look at that. We say cysteine, and we said that cysteine easily dimerizes. That's what we said in class. It easily dimerizes. Cysteine easily dimerizes. That it implies that Dimers. To dimerize means implies when two molecules implies when two molecules come together to form a single molecule. We say that in such a situation you say that a dimer has been formed. So when cysteine easily dimerizes, it forms a compound known as cysteine. Okay, so we'll quickly look at that. So two molecules of cysteine can come together to form cysteine in the following pattern, which is your COO minus plus your NH3 CH2 and what? SH. So this is one molecule of cysteine. Then it combines with another molecule as follows. Okay, so when these two molecules come together, this hydrogen atom here and this hydrogen atom here are given off in solution to form cysteine. When they give up in solution, that's where the dimer comes in. This X combines with this X to form what? A disulfide bond. That's very key. So this gives off, and it's a reversible reaction. Okay, they can naturally come back together again, break up in solution, and form their separate cysteine molecule. So this gives us 2 hydrogen plus 2 electrons, and what you have here is your COO minus your CH and H3, then you have your CH2, and of course S. This H here has been given off, so you just flip this molecule to form this disulfide bond, S coming right there, CH2, okay, and your amino acid is formed beneath, right? So you have NH3 plus and NH3 plus, then you have your COO minus. Now this bond that is formed right here is known as your what? Disulfide bond. It's known as your disulfide bond. So this way you actually have a molecule right here of cysteine and another molecule right here of cysteine, all connected by what? A disulfide bond. This bond can be broken in solution, and what is there, what comes up later, is that your uh, two atoms have hydrogen and two um, electrons is goes into the solution in presence of water, broken the disulfide bond, and again forming these two molecules of what? Cysteine. So this is cysteine. This is cysteine. And what is formed here is what? Cysteine. Alright? So we said that the cysteine amino acid oxidizes to produce two molecules. So clearly what has happened here is this. Worthy to note, what has happened here is that two cysteine amino acid, two cysteine amino acids oxidizes to produce two cysteine dimers. Okay, so that's for this forward reaction. Now in the reverse reaction, which is this beneath, for forward reaction that applies. For the reverse reaction, this is what is happening. So let me just like indicate it here. Forward reaction. In the reverse reaction, what happened is that a cysteine dimer that is formed having a disulfide bond, okay, a cysteine dimer reduces to produce two cysteine. Okay, it goes back to where it's coming from. Two cysteine what? Amino acids. So that is very clear. Two cysteine amino acid, and you could say this is what? Reverse reaction. Alright, so in the forward reaction which we saw, what happens is that two hydrogen atom plus two electrons are lost, which is a process of what? Oxidation. Okay, and that is what we saw here. This is oxidation taking place right here. They are given off, and we say this is what? Oxidation. Okay, why in the reverse reaction two hydrogen and two electron are what? Gained, and you could say this is the reverse reaction, and in this case this is what? Reduction. Okay, so that is basically it for some amino acids with special properties. Okay, so we also have some other amino acids that exhibit some special features, just like this cysteine. So it's not just the cysteine, we want to move right along to see others. Okay, so let me do the others in It's a purple. Be fine. So we have other amino acids such as your tryptophan, and what? Thyroxine. So from your classes of amino acids, you could easily know where this group of amino acids is coming from. And to some extent, you have what? Uh, sorry. Sorry. And to some extent, you have phenylalanine. Now, if you look at these three amino acid mentioned here, you could easily see that they belong to the class of amino acid aromatic amino acid. So they exhibit special features as follows. They absorb ultraviolet light strongly. They absorb ultraviolet light strongly. Okay, that's key. And that is because they have the aromatic group. Okay, because they have or contain the aromatic group. So it's a special features. Alright, and they have very strong absorption at about a wavelength. So they have strong absorption. Any question? Are you sure? Am I being heard so far? Okay, so they have strong absorption at about a wavelength. Strong absorption of what? Ultraviolet light. So important to note, at about a wavelength is normally indicated by lambda sign of what? 280 nanometer. Okay, and this explains why proteins absorb UV light strongly. Okay, it explains why protein absorb UV light very strongly. Okay, so when this wavelength is high, so thus it explains why proteins absorb ultraviolet light, that's what UV light is, strongly. Is simple. It is because of what? The tryptophan, thyroxine, and phenylalanine that is present. So the why is simple because of what? The aromatic amino acids such as because of the tryptophan, all of these are aromatic amino acid, your thyroxine, and what? And phenylalanine, phenylalanine, that is present, that is present in what? Proteins. That is present in proteins. So this gives us what? An analytical tool by which we can measure or what? Identify proteins. So it's a special features. So there are other special features of amino acid general. We've looked at cysteine, we looked at the aromatic amino acid ability to what? Absorb UV light. Alright. Then we have things like the uncommon amino acids. Okay, they are not common. Some are derived from common amino acids and are modified after the protein have been synthesized. So examples of these common amino acids, okay, is because of their modification, that's why you have them as common. There is either addition or subtraction from particular amino acid residues. Basically, uncommon amino acids are basically and mostly derived from existing amino acids. Okay, so examples of some uncommon amino acid that we have are, okay, so let me move up a little bit to have that. Alright, so examples of the some uncommon amino acids. Examples of some uncommon amino acids include the following. Okay, so you have 5-hydroxy, 5-hydroxy lysine. So we saw the structure of lysine not quite long. I think they are under the positively charged amino acid. This 5-hydroxy lysine can also be called your delta hydroxy lysine. So I gave you the structure in class. I'll quickly just do that here again. So the 5-hydroxy lysine, you have something like this. CH2, CH2, you know, lysine is made up of 4 methyl group from the amino acid backbone. So this is attached to the what? Amino acid backbone. Here you have your alpha carbon attached to your hydrogen atom, attached to your amino group, and of course your what? Carboxy group. So you could do this. Alright, so this is your alpha carbon here, and this is what? Your beta. This is your delta M, and this is your delta. Okay, now this one here, counting from this point, is 1, 2, 3, 4, 5. So you have 5 hydroxy. There's an hydroxy group here, so that's why it is called 5-hydroxy lysine, or what? Delta hydroxy lysine. Then another example, this is just one of them. Okay, another example right here of a common amino acid is what your um, omega carbon glutamate. Okay, and its structure can be given below. So you have this as a structure. Of course, you know glutamate has two um, carboxyl group. You have CH, and it's this CH now that is bonded to another carboxyl group right here, which is actually the attachment. And of course, you have CH2, and this is bonded to what your amino acid backbone, where you have your amino group attached, your hydrogen atom attached, and what your carboxyl group attached. Now this is alpha, beta, delta, and omega. So that is it structurally. So that actually closes our lecture for the amino acids. Okay, and we went right along to begin to look at DNA structure, RNA, DNA. I gave you an assignment. Please, in your interest, try to know that assignment. Read it up very strongly. Okay, it's not just an assignment, it's a continuous assessment for me, and knowing it will help you in your exam. So who can recall the assignment for me? So in that assignment, I think we looked at steps of protein synthesis and how protein is being synthesized. We looked at what is protein synthesis in itself, then how it is being synthesized. I think that's the last class. And we looked at the RNA as genetic material in virus. I'll pretty much stop there, and all of that are in your um, in the material that I think you have. So please, you run through that as part of your revision, the assignment in particular, and the last class where we look at the steps in amino acid. So that's we pretty close the revision time for today. Because of your exams, I don't want to go into much details of areas we could not cover, so that I don't get you confused. But I've pretty touched every area that we had lectures on, and at this time I'm open for questions. If you have questions, particular questions you are not clear about and you want me to answer, please now go ahead and type your questions so that we begin to look at them. Am I still being heard? done. I need questions.
No question.
Your voice is low. No, I need questions.
It was 7 before.
Only 7 people.
Are you gonna call me? I want to end the show.
They are asking, do they have any questions?
No.
Your voice is low.